Amatoxins are a class of oligopeptides found and produced in a few species of mushroom, notably Amanita phalloides, popularly referred to as the death cap. This mushroom is known as the death cap because amatoxins, and in particular alpha-amanitin, are highly toxic with an LD50 of approximately 1 milligram per kilogram. The toxicity of this molecule derives from its high binding affinity to RNA polymerase II. It binds at the bridge helix of RNA polymerase II and greatly reduces the mobility of the enzyme in translocating RNA in the process of the elongation of messenger RNA and the production of proteins. The lost mobility of the enzyme slows the translocation rate by 3 orders of magnitude, practically halting the functions of RNA polymerase II entirely. In addition to the halting of RNA polymerase II functionality, alpha-amanitin seems to trigger the production of many reactive oxygen species in trials in mice. The production of these oxygen species in oxidative species in combination with the halting of RNA polymerase II causes membrane degradation in the cell, which causes leakage of organelles into the extracellular matrix. Eventually, this leads to cellular processes of apoptosis, triggering cell death. Binding to RNA polymerase II is caused by a bicyclic structure with a thioether bridge between a cysteine and a tryptophan in the molecule, forming a tryptothionine. And the small size and rigid structure caused by this thioether bridge allows for a tight binding affinity in between the molecule and RNA polymerase II. This conformation binding affinity is also greatly increased by a beta turn at isoleucine 6, and the absence of any side chain at the proximal 5th glycine. Experimental trials which substituted these specific amino acids led to near total reduction of the binding affinity of the molecule. The molecule is produced by cleavage from a proprotein, which forms a linear polypeptide. This polypeptide is post-translationally modified by cyclization at the terminal protein to the initial isoleucine, followed by the formation of the thioether bridge, and then hydroxylated and sulfoxidated in order to greatly increase the binding affinity of the molecule. Symptoms of alpha-amanitin poisoning are primarily hepatic toxicity. This is because the molecule dissolves directly into the blood serum. In the blood serum, it is transferred to the liver simply by the fact that the liver processes most of the blood in the body. In addition to the proximity of the liver to the blood, the molecule is directly transferred into the liver by a shuttling protein, liver-specific anion transporter 2, which leads to rapid absorption. Within 36 hours, the molecule will fully be absorbed by hepatic tissue. The shuttling by liver-specific anion transporter 2 has led to the development of a few treatments for alpha-amanitin toxicity, including administration of rifampicin, silibinin, penicillin G, cyclosporin A, and cholecystokinin octopeptide, all of which block the transport of alpha-amanitin through liver-specific anion transporter 2. The high polarity of the molecule has also led to successes with cyclization of alpha-amanitin out of the blood with activated charcoal.